Hi, my name is Jessica. I'm a biologist and PhD student in ecology at the Marine Ecology Lab here in Brazil. And today I'm going to present my work on identifying coral bleaching hotspots, a predictive and functional approach. Corals are threatened by climate change as oceans are predicted to warm between 1 and 5 degrees Celsius on average by the end of the century. The most notable response of this threat is coral bleaching, a phenomenon that is becoming more frequent and severe over time that can reduce corals' thermotolerance, recovery capacity, and lead to declines in coral cover. Marine organisms are sensitive to temperature changes and can have their phenology and physiology altered by increasing ocean temperatures, causing range shifts through expansion or contraction of species distribution. Most sclerotinia corals have their physiological thermal limit between 18 and 28 degrees Celsius, and with warming, some regions might exceed this coral's upper physiological limit, becoming less suitable for corals, while other regions might increase their minimum temperature and become more suitable for corals to thrive. But corals could also acclimatize and adapt to these increasing temperatures, which is most likely to occur when global and local stressors are minimized. Therefore, identifying the most vulnerable areas to coral bleaching could inform actions to minimize local impacts and reduce threats from global stressors. Beyond identifying the most vulnerable areas to coral bleaching, looking at coral traits such as symbiont diversity, growth rate, and reproduction can indicate the potential resistance and resilience of vulnerable areas, indicate how these areas will cope with climate change. In this context, we aim to predict the outcomes of coral assemblage and find the most vulnerable areas to coral bleaching, defined here as vulnerability hotspots, under a high emission scenario. We used coral occurrence, coral cover, and coral bleaching data of 23 coral species across a 28 degrees of latitudes in the Brazilian coast between 1993 and 2017, collected from articles, reports, dissertations, and theses searched on Google Scholar, Web of Science, and university repositories, together with selected environmental variables for current time projections between 2000 and 2014. After selecting the best fit model and establishing the relationship between biological data and environmental variables in space under present conditions, we predicted their current distribution and future projections under a representative concentration pathway 8.5 climate change scenario using a Bayesian model. For each time period, we overlap the projections to find the vulnerability areas, using coral occurrence, coral cover, and coral bleaching as equal conditions to define vulnerability areas, in which a high vulnerable area to coral bleaching, vulnerability hotspot, will be those with high coral occurrence, cover, and bleaching probabilities, while areas with the lowest probabilities of coral occurrence, cover, and bleaching are the least vulnerable areas and might be considered as refugia. Regarding the functional approach, we used 9 coral traits related to resistance and resilience of the coral species to build a trait space of each region indicated by the different colors in the left map using a PCOA. Overall, our results show a high probability of coral occurrence, cover, and bleaching within the tropics, which can be seen in these 3 figures, where a high probability is represented by the reddish color, a low probability by the bluish color, and the transitioning zone is delimited by the dotted line. Well, these probabilities are likely to increase as ocean warm, where coral occurrence showed the greatest changes, increasing southward with warming beyond the 20 degrees south and decreasing in the tropics between 1 and 20 degrees south. While coral cover also increased beyond the 20 degrees south, there was almost no change in the tropics between 1 and 20 degrees south. Despite coral bleaching probabilities increasing overall at the coast with warming, the increase was at a smaller magnitude compared to coral occurrence and cover. Looking at this figure, which shows the transitioning zone between tropical and subtropical regions, we can see the southward increase in coral occurrence and cover, showed by the black arrow, that reveals new suitable areas for corals despite the coral bleaching probability increase. Several marine organisms are shifting their distribution polar in response to warming, and corals are likely to do the same. Shifts has already been recorded for the appropriate species in the east coast of the United States and in Japan, and predicted for the Brazilian endemic brain coral Mrs. Miller-Harty. Our future projections also indicate deeper offshore reefs as critical areas for coral occurrence and cover, which are indicated by the black arrow, which might also play a role as refugium, as deeper sites suffer less from anthropogenic impacts and warming ocean temperature. So, despite not having a consensus about the deep refugia hypothesis due to low connectivity between some shallow and mesophotic reefs, the Brazilian coral species are depth generalists, where the same coral species often occur in both shallow and deeper sites, such as Montastic cavernosa. Therefore, these reefs with depth generalist corals may favor the deep refugia hypothesis in response to warming. By overlapping the three projections, we found more than 80% of the area under intermediate vulnerability, only 3.13% as the highest vulnerable areas, the vulnerability hotspots, and 12.6% under a low vulnerability, the potential refugium. However, with warming, the refugia area could experience a 50% reduction as they increase in vulnerability, which might challenge its role as refugium. The vulnerability hotspot area, indicated here by the transparent line, includes the largest and richest reef system in South Atlantic, the Abertis Bank, which is worrisome as it is a unique site that comprises high coral cover and endemism level up to 30%, and diverse reef morphologies. Well, the positive side is that the Abertis region also comprises several marine protected areas that may mitigate local stresses and enhance the ability of corals to cope with rising temperatures. In addition, corals in the Abertis Bank show high recovery capacities after bleaching events, except for hydrocorals, likely resulting from a combination of minimized local impacts and high turbidity in the region, which can reduce intense light incidents in the water, helping corals to recover from bleaching. When we look at coral traits to evaluate the potential resistance and resilience of vulnerability regions, we found that the most vulnerable region, which is here indicated by the dotted circle, comprises the greatest species richness and the largest functional trait space, which could indicate high resistance and resilience. Therefore, despite being the vulnerability hotspot area, coral traits such as fast growth rate, reproduction, and semiodinium diversity can help to cope with climate change vulnerability in the region. So what we did? Here we modeled coral occurrence, cover, and bleaching data to current time and future scenarios under a high emission scenario using a Bayesian approach, and overlapped the three projections at each time period to find the vulnerability areas. We found newly suitable areas for corals in subtropical and mesophotic reefs, which may act as refugia. But coral bleaching will also increase in the future, but less intense, but still might challenge its role as refugia. The vulnerability hotspot is concentrated between latitudes 13 and 20 south, which comprises several reef habitats within the most diverse reef system in South Atlantic, the Abertis Bank, a region that, despite being the most vulnerable area, is protected from local impacts and has the largest species richness. By using a trait-based approach, we found that the vulnerability hotspot region also has the largest trait space, which could indicate its resilience and resistance potential. In conclusion, we recommend marine protected areas to protect the vulnerability hotspots, current and future refugia, and new MPAs in between them, acting as corridors to favor a possible range expansion of coral species with climate change. If you need more details, this study was recently published in scientific reports, and you feel free to contact us. I'm grateful for all the Brazilian financial support involved in this work, for the ICRS travel grant, and the opportunity to share my work here at this event. Here you can find all my contacts and the DOI of the recently published paper. And thank you for listening.